Hi, I am Dr. Loral Showalter, and I am doing what I love, fighting cancer.
Today, Lori is going to show us how to make a mouse version of a dendritic cell vaccine that we use in experiments to perfect our approach. We keep these cells frozen away in liquid nitrogen in suspended animation until we need to use them. Those small plastic vials with the orange caps contain the cells that Lori took out of liquid nitrogen. As they thaw, she carefully removes the cell suspension with a pipette and transfers them to a larger tube. The cells were stored in a chemical solution that keeps them alive while frozen, but can't be there when the cells are cultured at room temperature. Lori will dilute out these chemicals by adding a large volume of red liquid in that big bottle you see in the hood. The way we separate cells from the diluted freezing chemicals is to centrifuge them. The cells will sink to the bottom of the tube, and the diluted freezing chemicals will remain on top. The cells have spun down now. Lori is using the sterile pipette attached to the vacuum line to suck out all those freezing chemicals. You can see that big pellet of dendritic cells at the bottom of the tube. Now she'll resuspend the cells in some clean, fresh culture medium. The next step is to count the cells. We need to know how many cells we have in order to do the experiment. Here Lori is using the great new microscope purchased for us by Pennies in Action. The video camera captures what Lori is seeing. She counts the cells within that grid on the monitor to determine their numbers. Now Lori has the dendritic cells distributed into those small plastic dishes. She is going to add synthetic pieces of the HER2 cancer protein as well as biochemical signals of infection to trick the dendritic cells into thinking that cancer proteins come from a dangerous infection. That way, when they're placed in the body of the tumor-bearing host, they will rally the immune system to attack the cancer as though it were an infection. A few hours in the incubator, and the cells will be ready to inject. We recently did a study where we combined vaccines with statin drugs, normally used to treat high cholesterol. Statins can affect how cancer cells use certain fats, and we had reason to believe that they might amplify the effects of vaccination. And we were delighted to find out that it worked. This graph shows tumors growth over time for untreated mice, mice treated with vaccine, simvastatin, or both at once. As you can see, single treatments had little effect, but when both vaccine and drug was given, the tumor growth slowed almost to a halt. Thanks again for your support. Without you, we could not envision a day when cancer will be eradicated, not only in people, but also in our companion animals. Help us continue to fight the good fight.